let us start. Let us start the second part of this evening day, so to say. And it's a great pleasure for me to introduce our next speaker, Erwin Frey, and welcome him to the evening lecture. It is a special case for me since I know him already over a longer time, but let me first congratulate him for being awarded last year by the Max Planck Medal, the highest award of the German Physical Society, for his fundamental contributions to theoretical biophysics, in particular to intercellular transport, biomolecular processes, and pattern formation in complex systems, the subject of the lecture he gives at this year's evening lectures. Secondly, the nice thing is that Erwin and I are related, as we are leaves on the same family tree, which was discovered by Elliot Montroll, which he described 1982 at the conference on random walks and their applications in the physical and biological science at the Loyola Institute under the title Vienna School of Statistical Thought. Montroll noted a remarkable large number of pioneers in the evolution of the statistical style of thought in modern physical and indeed biological science may trace their family tree back to the Vienna School. In this tree, he also connected to Smoluchowski, who 1897 found no position open for him in Vienna, but at the University of Lviv. And Smoluchowski research on Brownian motion, fluctuations, and critical opalescence, basic aspects of stochastic processes, such as calculation of first passage times, and the development of our understanding of non-equilibrium processes. It is closely related to the topics covered in the Ising lectures. Erwin received his doctoral degree in 1989, supervised by Franz Schwabel with a thesis on the static and dynamic critical behavior of dipolar ferromagnets. From 1990 to 1992, he was postdoc at Harvard University, and then he returned to the TMU, where he worked as a research associate until 2001. In 1996, he completed his habilitation at the Technical University in Munich with a thesis on the statistical physics of fluctuating manifolds. From 1998 to 2001, Frey was a Heisenberg Fellow in the German Research Foundation and a visiting lecturer at Harvard University. At the same time, he was visiting professor at the Ludwig-Maximilians-Universität Munich, and from 1999 to 2000. In 2001, he was appointed full professor of physics at the Freie University of Berlin. From 2002, he took over as a head of the Department of Theoretical Physics at the Hahn-Meitner Institute. In 2004, then, Frey was appointed to the chair of Theoretical Physics, Statistical Physics at the LMU, Ludwig-Maximilians-Universität in Munich, a position he has held ever since. Erwin, the screen is yours.
Thanks a lot, Reinhard. That was a very nice introduction. And of course, these shoes that you were painting, they're way too big for me. But still, it's a great, great honor. So thank you, dear colleagues, for inviting me to the Ising lectures. I'm happy to give the lecture, and I hope I can entertain you with my recent interest in self-organization and emergence in biological systems. And as usual, I mean, I'm happy if you interrupt me along the way. Don't wait till the very end before you ask any questions. It may be a field which is not your mainstream field because it touches upon biology, but I try to connect it as much as I can to theory. Hope you can hear me well. It's all fine, right? Yeah? Okay.
Yeah, we do hear you.
It's okay. Yeah, we hear you. Very good.
Good.
Everything is working.
So there's two topics I would like to talk about. One is self-assembly of biomolecular structures, and the other one is protein pattern formation. For today, I'll focus on the second topic, protein pattern formation, and basically ask the questions, how do protein patterns self-organize in space and in time? And tomorrow, I'll talk about a topic which is about the self-assembly of biomolecular structures. And there I would like to talk a bit about complexity, time complexity. And as you will see tomorrow, there's even critical phenomena when it comes to the process of self-assembly. But as said today, let me talk about protein patterns. What I mean by that, here is an E. coli cell, a movie of an E. coli cell. This is about 1.5 or 2 micrometers in length and a micrometer in diameter. And you see these oscillations, they go back and forth between one cell pole and the other. And these are oscillations of proteins that are just a nanometer in size, and they do diffusion, they do reactions. And as a consequence of these diffusion and reaction processes, they bounce back and forth here. And there's a task for that process. If we average the oscillations over time, you get a profile which is maximum at the side and minimum in the middle. And that's where then you have the formation of a protein ring that pinches off the E. coli cell and makes 2 faithful daughter cells. There's also budding yeast. There's another cell which has a different type of cell division. The way it works is it forms a bud site by another type of protein pattern. And at this bud site, you then get a bud and a new offspring of the cell. And finally, when you look into higher organisms, like here a worm, C. elegans, there you see that patterns form in order to define cell polarity. So these are beautiful mesoscopic phenomena of collective pattern formation, and linked to these mesoscopic phenomena ranging from a micron to a multi-micron scale, there is molecular processes. And you don't have to look at the details of all of that now, but basically what it is, you have a membrane, you have a bulk, the cytosol. There's lots of reactions going on in the membrane. There's reactions going on also in the cytosol, but much less. And you have constant cycling of proteins between those two compartments. And let me explain that a bit for the first example, so for these min oscillations, because it's simplest to explain. It's basically 2 proteins. They have names. They're called min D and min E. And there is a quite generic design of the biomolecular reaction network that you can see here. So let me start from the min D ADP. That's the blue circle here on the right. This is an inactive molecule, and it's activated by what's called a nucleotide exchange. This is a chemical step that conveys chemical energy to the system and makes the passive or inactive molecule with the ADP bound to it to an active molecule with the adenosine triphosphate bound to it. Now, once this molecule is activated, it changes its properties, and the main property it changes is it's now able to bind to the membrane. So as you see, there's an arrow showing that this protein can bind to the membrane. And then once it is bound, there is a feedback mechanism. And this feedback mechanism recruits more and more min D proteins to the membrane. And if that would be just the only feature of the reaction network, you would have accumulation of these min D proteins on the membrane. That would certainly not give any kind of oscillation. So what nature has designed is that it has a second type of protein, with the name min-E. It's also recruited to the membrane by the min-D protein. They form a dimer. And when they form a dimer, this min-E protein is actually hydrolyzing the min-D protein, so making it from the ATP form into ADP form. And then the protein can no longer hold on to the membrane. Both of them fall off back into the cytosol. And as you see, the whole cycle here restarts. And that's quite generic. So you see there's a driving force, a chemical energy that's put into the system. There is a cycling of fluxes between different states, monomers, dimers, active, inactive, and they're cycling between the cytosol and the membrane. And this is really, I mean, in the heart of Kolmogorov and others, you have cyclic fluxes. That means you have broken detailed balance. And because you have broken detailed balance, you have irreversible processes. And that means these systems can have non-trivial behavior, which you can't have in a thermodynamic system where detailed balance would not be broken. Okay, so that's the heart of these systems. They all have broken detailed balance, and thereby they have non-equilibrium fluxes of proteins between states and between compartments. That will be a major theme, as you will see. All right. What would you like to know when you study such a system? There's many questions you can ask. There's questions you can ask that come from biology. There are questions that you can ask that come from biophysics, and there's questions which are much more conceptual and theoretical. So one question, which is a biochemistry question, but quite important, is what's the role of the reaction network? And that's by no means a trivial question, because the network, which is shown here on the slide, is a network which you infer from biochemistry data, but there is at present no way to really check in detail whether this is the exact and the right biochemical reaction network. And if you're trained in critical phenomena like myself, you would think, oh, well, details don't matter, so don't worry. You can get away with a simple model, and that will explain the data. But you'll see it's a very subtle thing here. Details in biology do sometimes matter in a very interesting way. The other question, which is quite interesting, is whether these patterns are able to sense geometry. I guess you all heard about the question, can you hear the shape of the drum? The answer, of course, is yes, because the boundary conditions determine the selection of the mode spectrum, and that's what you hear when you listen to a drum. This here is a nonlinear process, so those are not linear phenomena with a superposition principle. These are nonlinear waves. But as you see from this movie on the right-hand side, this is data from K.S. group. If you change the aspect ratio of the E. coli cells, you get pole-to-pole oscillations, you get side-to-side oscillations, and the structure, which has almost even aspect ratio, has a pattern that looks very much chaotic because it changes from one side to another side and even shows some kind of circular motion. So that means the geometry is a design feature of these kind of patterns that really feeds into the selection of the pattern. The third question, which is of course dear to my heart, is, are there some general universal principles that you can figure out by looking at these reaction networks, and can this somehow contribute to a deeper understanding of non-equilibrium physics and non-equilibrium thermodynamics? So here is a blow-up of the general problem that we face. You have a complicated geometry, which here on the right-hand side is the spherocylinder describing a E. coli cell. There's a nucleoid, which is the DNA inside of this E. coli cell. There's the cytosol, that's the bulk, and there's the membrane. So you have different geometric compartments. And in these different geometrical compartments, there's different diffusion coefficients. The diffusion coefficient in the cytosol is usually very fast. So to give you a number, it's about 10 micrometers squared per second. If you go on the membrane and ask about diffusion on the membrane, this is by orders of magnitude slower. It's something like 0.01 micrometer squared per second. And then what you have is you have a diffusion reaction equation on the membrane, with diffusion on the membrane and reactions on the membrane. You have the same type or similar type of diffusion reaction equation in the cytosol. And linking the two, you have a reactive boundary condition. And this reactive boundary condition basically says, if you want to conserve particle number, then any kind of gradient in some concentration that creates diffusive fluxes has to balance the corresponding reactions so that you don't lose or gain particles as you go from the boundary between the membrane into the cytosol. All right, so let's look at a molecular model. This is the yeast system. You can use the model that I showed you early on to simulate the system. This is now the simulation. And you see the model does what it should do. It shows the same kind of phenomena as you see in biology. But that's hard to interpret, hard to understand in terms of first principles. So I would like to go with you through to something I think Ising had done also when he wrote his thesis, to make the problem as simple as possible, but not too simple. So let's go to a minimum set of variables. And this is really as simple as it gets. You just think of the system as a sphere, and you have a single protein, right? Just one protein. And this one protein can be in two states. It can be either on the outer shell, on the membrane, can be membrane bound, or it can be in the cytosol. So a two-state model and one species. The geometry that I showed you is still too complicated, so let's make this 1D for now. And then when you write down the equation for that, it's a very famous set of equations, actually. You say that the temporal derivative of the membrane concentration M, the temporal derivative of the cytosolic concentration is reaction terms, which I call F and G, and both of them have some diffusion term on the right-hand side. And F and G can encode any kind of reaction that you like that somehow describes this nonlinear system. Well, but this equation, right, this is a famous equation written down by Alan Turing, and we heard about Turing machines now we hear about Turing patterns. This equation that you see here is a very general mathematical question, but for the purpose of what I'm doing here, it's actually wrong. It's wrong in a subtle sense. If you look at the protein mass, N, it has to be conserved. There's nothing that can create or destroy any kind of proteins. So N must be a constant of motion. So as you add up the two equations, the reaction terms have to cancel. So that means that you have to have a set of equations where the function G is identical and opposite to the function F. So you have minus F here in the second line. That seems like a minor modification of this set of reactions, a set of equations by Turing. But as you will see, this has a very, very important implication. I try to make a connection to statistical physics also to explain to you why this is such a important modification of the equation of motion. If you rewrite this equation of motion, you can add it up, right? And as you add it up, the reaction terms obviously cancel, and what you get on the right-hand side is just diffusion terms. And when you rewrite the right-hand side in terms of a potential eta that's defined by the left-hand side, you get a model, for those that know it, this looks very much like model B. Just has no noise, but it has a constant, a mobility, times a Laplacian of a potential. And if it were to be model B, then the potential eta that you see there had to be a chemical potential, and you would need to derive it from a free energy. So this would, you would need this to be a functional derivative of a free energy function. But it's not. Here, you don't have a thermodynamic problem. You have a non-equilibrium dynamics problem. So when you do the math, you figure out this potential, this mass redistribution potential, that's how we call it, is a dynamic problem on its own right. It has an equation of motion with a diffusion term, but also with a reaction term, and this F tilde is basically the same function as F. Okay? So that's the difference between a equilibrium and a non-equilibrium model, that you have a non-equilibrium potential that is a dynamic quantity and not a quantity slaved by the free energy function. So now let's go from equations to pictures and show you how you analyze this and how you can think about such a mass-conserving diffusion reaction system. So let's consider a box, a well-mixed box, and let's think about the system such that diffusion is super fast, so you can neglect diffusion and just look at reaction terms. And as an example, I put down here a reaction term which contains attachment with a function A that's proportional to the concentration in the cytosol, and minus, sorry, this is detachment, which is some function times the membrane concentration, since that's the molecules that detach from the membrane. The equations are as simple as you can think. It's temporal change of M and C are both given by F, and there's a constant of motion, which is the total density. And now let's analyze this in phase space. So let's draw concentration on one axis and concentration on the membrane on the other axis. And this line that you see now is the null line, so F equal to 0. That means that's the set of equilibrium points, of chemical reactive equilibrium points. That alone doesn't determine the fixed point, the steady state of the system, because it's a whole function. So what you need in addition is this constraint that the total mass is conserved, and that defines a simplex. And on that simplex, let's assume that the fixed point is stable, then all the flow is on that mass conserving simplex towards the fixed point. So you see, the fixed point itself is a function of the properties of the reaction. So it's on the null line. So it will depend on all the reaction rates that you have in the system. But importantly, it's also a function of the total protein concentration, N. So now, as you change the total protein concentration and move it to the right or left, you will change the equilibrium point. And that means, right, that the control parameter that determines the system is not just the reaction rates that you can tune, but also the density of proteins that you have in your system. And that's key because in thermodynamics, when you think about control parameters, you think about something external. You change a temperature, you change a magnetic field, you change some degree of other variables. Here, the control parameter N becomes later on, when you talk about dynamics, a field that's varying in space and in time. And thereby, you can have different behavior at different points in time and different points in space, and symmetry breaking that's controlled by a control parameter field and not a control parameter itself. All right, I've said all this, and I think another important point to make is, if you look at this construction, it's very much in the spirit of a geometric picture of nonlinear systems where details actually don't matter, and all that matters is what kind of topology or geometry the flow of the differential equation has here in phase space. So now let's move on from a single well-mixed compartment to space. And now let's think of my E. coli cell as composed of two compartments, one on the left pole, I call 1, and the other one on the right pole, I call 2. You have not just reactions, but now you have diffusive exchange between the two compartments. And let's say the diffusion in the cytosol is the dominant one because it has the larger diffusion coefficient. That means then that the equations have an additional term, and especially the total concentration N1 and 2 in the two compartments now change over time in an opposite way. So one's gain is the other's loss. Now let's start how patterns emerge. So I start from a uniform concentration where in the compartment 1 and compartment 2, you have the same concentration, N1 and N2 equal N bar. And now imagine that I put on a little perturbation. That means I increase the mass in the compartment 2 and I lower it in compartment 1. So you here in this graph at points 1 and 2 in phase space. Well, now that you have this change in mass, the system will respond because there's chemical reactions. And what I'm doing now is I'm kind of doing a timescale separation where I say, imagine that the reactions are now much faster than any kind of diffusive process. And if that's the case, then it will pull the system along the mass conserving planes in compartment 1 and 2 towards their respective fixed points on the null line. But now, as you look at the geometry of that, you have increased the concentration in the cytosol. You have moved up on the left and you've moved down on the right. So you have a gradient from left to right in the cytosol. Well, and the gradient implies a diffusive flux. A diffusive flux implies mass transport. And that means you're transporting mass from 1 to 2. But that then increases the already existing difference in mass between the two compartments. So you have positive feedback that you kick off, and this positive feedback amplifies the mass difference between the two compartments. And as you look at the geometry of the phase space analysis that I just did, this hinges on the fact that the slope of the null line in orange is negative. If the slope would be the other way around, my argument would be opposite, and the cytosolic flux would not be from right to left, from left to right, but from right to left. And that would then attenuate an initial perturbation and drive the system back to the original homogeneous state. So what we see here is a geometric picture for the emergence of a pattern-forming instability, and it's simply linked to the slope of the null line that we have in the system. Well, now I have been simplifying things a bit, okay, and the polar state that emerges from that is one which is here kind of flat. But if you add the diffusion here on the membrane as well, my argument would be a little more complicated, but hard to explain in a short talk like so. And what you then get is that the criterion for instability is not that the slope of the null line is negative, but the slope of the null line needs to be steeper than a slope which is defined by the ratio of the diffusion coefficients on the membrane and in the cytosol. And this is really as simple as it gets. This is the simplest, the generic mechanism for protein pattern formation, for the formation of polar zones in the E. coli cell, and for the formation of a polar cap in the yeast cell. And as you've seen, it's driven by redistribution of protein mass, linking to what I've said at the beginning, that the control parameter mass is an essential control parameter in the system and determines the physics of the pattern-forming dynamics. All right, so let me show you how this plays out when you simulate it. This here shows two things at the same time. On the left, you have a real space plot where you show the membrane density and the cytosolic density, and on the right, we do have a phase space portrait. So here you have a small modulation of a membrane density. There's a small modulation of the cytosolic density, and that means you have a cloud of points that you see here in the phase space. And each point here in phase space corresponds to a point here along the real physical space of this one-dimensional model. And now as I run the simulation, you will see the patterns forming in real space and attractors forming in phase space. And note what kind of attractors you have. So at first, you have an instability due to mass redistribution. Then the system settles on this line, right? And then it stays on that line. It's a very strong attractor of dynamics. But there's slow dynamics happening along this line. And the slow dynamics, as you see in real space, leads to coarsening, and in the end, to complete separation of density, low membrane density on the left, high membrane density on the right. Well, if you know phase separation physics, the phenomenology of liquid-liquid phase separation is quite the same thing. It also starts with binodal decomposition, pattern formation. There is Ostwald ripening described by LSW theory, and there's coarsening, and in the end, you have complete phase separation. This is the same phenomenology here, but the reason is different. It's not a system driven towards thermodynamic equilibrium, but towards a non-equilibrium steady state. And this non-equilibrium steady state has some hallmarks, some features. One is that you are on this subspace in blue, which is given by a line that has the slope of the ratio of diffusion coefficients. That's we call diffusive flux balance space. And it's basically saying that diffusive fluxes on the membrane and the cytosol have to cancel. And this diffusive flux balance then defines this line in phase space. You might also notice that the area here and there, so between the NARC line and this flux balance subspace, it looks the same. And this is indeed a feature. You can show that if you integrate over all reactions in your box, the total amount of reactions has to conserve mass. And this reflects in the fact that the areas that you see here have to be approximately equal. And that's quite akin to a Maxwell construction, but it is not a Maxwell construction. It's a construction that is based on reactive equilibria and diffusive flux balances. So it's a non-equilibrium generalization of such a construction. So once you have the structure in phase space, you can have a discussion of the hallmark points that you have there. There is the two plateau values given by the intersection with the NARC lines, the green points. There's an interface in between. And then you can construct from this geometric picture the whole phase diagram. And this whole phase diagram then looks like this. You have on one axis the diffusion constant ratio, on the other axis the density. And this, what you see here is a orange regime, which is the one driven by the linear instability. But there's a wider regime like a spinodal and binodal regime, where you do have patterns that form only if you kick the system hard enough. So that means if you have some strong enough initial perturbation that gets you over a barrier. Okay. So that's the main outcome of this analysis of a two-component system. And if you like, you can read more about this in some lecture notes I wrote, and also in the longer article together with Fridtjof Browns and Jakob Haladek in Physical Review X some time ago. And let me summarize the key insights that you should have gotten from this analysis. So you can rewrite or redraw the diffusion reaction systems in phase space and fully understand what's going on by a geometric picture. This geometric picture gives you answers to two very basic questions. One is, why do those proteins self-organize into concentration patterns? And the second one is, what determines the pattern's properties? Well, the why question is because there is an instability. The instability is one where you redistribute the protein mass between the different compartments of your system. And what determines the patterns is the local equilibria and the diffusive mass transport that changes these local equilibria. And based on that idea, you can actually really construct what we call a local equilibria theory, where the conserved protein dynamics drives the transport. And it's really the picture I tried to allude to before. You have the density acting like a control parameter field, this control parameter field breaks symmetry in different parts of the system in different ways, meaning there's different equilibria, local equilibria, as a function of this dynamic field. Now, let me come to a question which is different from that. So this was about principles and a simple explanation of the basic phenomena underlying pattern formation. The question I would like to address now is how molecular really is molecular. So how many details do you need to really understand the min pattern in E. coli cells? Not just the principles that lead up to some pattern formation in mass conserving systems. No, really in the concrete situation of a E. coli system, what are the molecular features that actually do play a role in defining the patterns that you see and its robustness to all kinds of external perturbations. So here is again the network, and on the left you see the corresponding finite element simulation. The finite element simulation shows the features that you expect from such a system. It shows pole-to-pole oscillations. And there is an insight that you can gain, which is general, which is beyond the simulations, namely that the number of proteins that you need in order to make this work has to be partitioned in a certain way. You need that the min E proteins, the antagonist kicking the min D off, they need to be less in number than min D. Well, why? If it would be the other way around, then the min E proteins would be abundant in number. So as the min D binds to the membrane, they would be kicked off all the time, and they couldn't really form a high concentration profile to begin with. So to begin with, the min E concentration should be low, such that min D can actually accumulate in enough numbers on the membrane before kicking it off and giving rise to the pattern forming process. And that's a very clear prediction, right? This is something where you can put your finger on. If this agrees with the data, you're happy. If it disagrees with the data, your theory is actually wrong. So let's test. And that's the beauty of this field. You can not only study these biological systems in a living organism, you can also study it by reconstituting, so recreating the system outside of a cell. So what people do, and this is basically the work of Petraschwili and Kees Dekker and others that have been working on reconstituting these biological systems, they take the proteins out from the cellular environment, put them in the Petri dish. On the bottom here, you have a membrane. On top, you have a fluid. And there, into this Petri dish, you just put the two proteins, min D and min E, and nothing but that. Of course, you have to add ATP. Otherwise, there's no fuel running the chemical reactions. And then Petraschwili and Martin Lohse, when he did his PhD in Petraschwili's group, they found these beautiful phenomena. They found that the min system shows spiral waves in the context of such a reconstituted in vitro system. Those are not oscillations back and forth, but they're related, and they indeed help us to understand the system in much more detail, because these experimental setups are easier to control than a biological setup. You can control concentrations, you can control in principle also the membrane properties, and you can control and manipulate the proteins in various ways. So here's the data. Simon Kretschmer in the group of Petra sometime later varied the min E to min D ratio. This is quite a shock if you look at it. You see patterns, not just when min E is less than min D, but also for the range of min E being much more abundant than min E. So that clearly shows that the original theory is wrong. It doesn't apply. It's just not fitting the data well. It's a qualitative result that we miss here. And the data show something very different, that the patterns are much more robust than you expected based on this simple model that I showed you. All right, so that made us think. And what you do then in those cases, I think what you should do in those cases is that you carefully study the biochemical literature and ask yourself what kind of information is available about the biochemical properties of these proteins. And what you find, and then make this long story a bit shorter than it actually is that the min-E protein itself is a switch. This min-E protein is not just a fixed conformation protein. It can actually open and close. If it's open, it has a membrane targeting sequence with which it can bind to the membrane. If it's closed, it cannot. Okay? The min-D switches this conformation of the min-E. So as min-E comes close to the min-D, it opens up, and as it opens up, it can bind to the membrane. What's kind of funny is when you look at this reaction scheme, is that the reactions happen on a very, very fast time scale, much, much faster than the minute time scale of the oscillations back and forth in the E. coli cells or the pattern dynamics that you see in the reconstituted system. So it wasn't clear to us whether such a fast process could actually change the behavior, the macroscopic behavior of the system. After all, when you taught about timescale separation, what you usually would say, if there's a fast process, much faster than the phenomena you observe, you should integrate out that fast process and deal with it effectively. But we didn't. And Jonas, then, the time in the group, didn't. So we looked at the model in its full glory. And that's the model. So you have not just one conformation of the min protein, but you have two conformations of the min proteins. And it explains the data quantitatively. And to make to illustrate that to you, the mechanism behind it is that this latent min-E, which is not able to bind to the membrane, this serves as a kind of buffer for high min-E concentration. You can have a lot of min-E, but if they're not active, they don't matter. So you have a buffer. And the min-D just recruits the amount of min-E proteins that it needs to run the process. So that explained the data. And then the critical test was if the experiments would not now be able to
Sorry, there is now some problem with the
Yeah, seems he is
connection.
frozen. His screen is frozen, yeah.
See, also, he wanted to say something.
Professor Frey's microphone.
I don't know what happened. Somehow got kicked out.
Oh yes, there you are.
Let me try again. I think that's where we left, right?
Yeah.
We see
Yeah.
it. Yeah. Now
Good. So let
the
me.
second part is visible.
So you see it now. So that's where I was kicked out, right?
Yeah, a little bit
Good.
earlier.
Okie doke. So we have this extended model with the min-E switching between the two states. And what I was explaining is that the min-E being able to switch between the two states gives a buffer, and this buffer then allows higher min-E concentrations to form patterns. So the critical test for that is that the experimental colleagues, in this time Simon Kretschmer in the group, was able to make a mutant. And this mutant was one where the min-E protein no longer was able to open up and close. It just stayed in one conformation. And then that pattern forming system with the min-E switch disabled, should show the phenomena of the first model that we had, namely that the patterns are only allowed if the concentration of min-E is less than min D. And indeed, as you see here, this beautifully shows that this is actually the case. So that's a beautiful confirmation of our theory and explains that the detail of the mini switch is important, and in particular, it's important for the robustness of the pattern forming system to show patterns over a very, very broad range. All right, now, this was a in vitro system, so a biologist would tell you that's fine, that's nice that you can do this, but what about the actual living system, an E. coli bacterium? Can you do the same thing? Can you do a phase diagram in a living system? Can you change the concentrations and map out what kind of patterns you get in the actual bacterial cell? I didn't really believe that that would be possible, but I guess it was something like 5 or 6 years ago. I met Suk-Yun Yun at UCSD in San Diego, and we were chatting and discussing, and he kind of took the challenge. He said, I can do this with my student, I can make mutants, I can really tune the min E, min D concentration. And it took quite some time. It was published just very recently, 2 weeks ago, basically, to get the phase diagram in the living system. And here is the phase diagram. On the one side, the experiment, and on the other side, the theory. Again, you see the axis, the min E concentration, the min D concentration. You see there's a very broad regime in color, green and in blue, that shows pattern formation. So it's the same kind of robustness that you see to variations in the concentration as in the reconstituted system. And on the right, you see a quantitative theoretical model from our group, from Hendrik Weyer, and it matches the data in a very beautiful quantitative way, not just concerning the range of patterns, but also the type of patterns. On the left, when you vary concentration, in the experiment, you see transitions between standing and traveling waves, which is indicated by these camographs. And on the right, for the theory, you see the same kind of transition between standing and traveling waves. So you do have a quantitative model. After many, many years of study, we were finally able to really have a quantitative description of the living system. So the main point I want to make is that you can combine theoretical studies and experimental model systems in the living system, also reconstituted, to arrive at a full quantitative model that has explanatory value for the biological process and is really of biological relevance. But question you can have is there novel non-equilibrium physics? And now, Reinhard, I don't know how much time I have left, so I want to gear a bit. You're muted. I can hear you.
Yeah, 2 minutes in fact, but we can prolong, I think, a little bit. 5 minutes.
Okay, then I start the story and I'll start the story a bit, but I then continue again tomorrow because I have time for discussion tomorrow again.
Okay, or should we start with the discussion now?
Yeah, I think I can postpone this to tomorrow because that's a good point of stopping here, because it's saying that theory gives a model that agrees with data, and now the next question would be, is there now cool, interesting non-equilibrium physics, and that's going to be a story for tomorrow.
Yeah. So I asked the audience, are there questions? May I start with just the last transparency you showed?
Yeah, can go back to that.
Yeah?
Yeah.
So what I see, I mean, your theory is very regular.
Oh, no, this is just the number of grid points that you have, because we can't do the analysis over all category details. Also, so the points.
So you have only two visible structures, and it seems that it's more complicated on the experimental side a little bit. So is this
Yeah.
maybe due to some, well, you might have some errors in you have another material or something like that?
Yeah,
or-
so the level of agreement between theory and experiment in biology cannot be, at the
Perfect.
percent level.
Yeah.
That's not what you're looking for. So the main point to take home is really that the regimes are similar and up to, say, 10, 20 to 50%, the boundaries of the phase diagram agree, and the patterns are in the right regimes of the corresponding patterns in the experiment in the theory. We have done a more sophisticated analysis than what I showed you here on the screen. So the experiments have measured the wavelength of the patterns. They have measured the oscillation period of the patterns. And then you can also quantitatively compare the wavelength and the period of those patterns. And that also agrees much better than the phase diagram actually to very high accuracy. Of course, there's some deviations everywhere, right? And you can attribute this to many factors. The membrane may have some inhomogeneity that we don't account for. There might be some features of the molecular processes that we have not accounted for, and. So this is not a strict model where I could infer the model one-to-one from some data, do a simulation and compare with experiments. The model itself is a matter of debate.
Okay.
And at the moment, this is the best model we have. It's the best agreement that we could reach up to now.
Yeah. Thank you. I see, Yuri, you have a question, please?
Thank you very much for your talk. At the beginning, beneath these big questions you put to the nature was what is the role of the network? And you told us that you are training critical phenomena where is some universality and this is washed away. And the network was not mentioned too often during your talk. Does it mean that you are going to do this or
yeah, what I mean with network is this, right? So
yeah, the
if
network
we
of
go,
reactions, right? Yeah.
Yeah, so when you go here, this network here, we added, the original network didn't contain the latent and the reactive min-E, the two conformations of the min-E. Okay. And so this feature of the network, this detail of the network is actually important, despite the fact that it's a fast process, orders of magnitude faster than the pattern that you see. And that's surprising, right? Because this is something which you could consider as not important, not relevant, because you could adiabatically eliminate a fast process, but you can't. And the reason, so this is what I mean. This is what I mean by details do matter in biology, in particular here. The role of this switch is also clear from the data that you see. So with the switch, the pattern is robust. You can vary concentration of a wide range versus a small range. So in terms of the function of the switch, this switch is the module, the element of the reaction network responsible for robustness of pattern formation in E. coli cell. If they would not have that, the pattern would not be robust. We would lose a very important biological feature of the network. And that's what I meant. There's other modules. I didn't talk about them, and I most likely don't have time. Another question that remained open was, what's the wavelength of the pattern in the in vitro system? So in the system that you see here in Petra Schüller's experimental data. And the in vitro wavelength is considerably different from the wavelengths that you see in the living system. And for a long time, we didn't know what the reason for that is. And now we know, now we found very recently that in addition to this min-E switch between two states in the cytosol, the min E after hydrolyzing the min D also hangs on the membrane for a while, and only after a while it detaches. And this additional element of the biochemical reaction network is the module that's then responsible for the wavelength that you have in the pattern forming system. So the way I like to think about these biochemical reaction networks is that you may be able to dissect them into certain modules that have specific macroscopic roles, like forming a pattern, making it robust, and defining the corresponding length scale of the pattern.
Thank you.
Okay, thank you. Are there other questions? Would you raise your hand? No, I don't see anything, but may I ask, so just to look at your results, it's a non-equilibrium system.
Yeah.
You have to put energy into the system.
Yeah.
And if you change the amount of energy you put into your system, then the pattern changes.
Yeah.
So is this how, so you have a range of energy you have to put in that something is working?
Mm-hmm. That is correct. So the energy in the system is the ATP, so the adenosine triphosphate, and the conformational change from triphosphate to diphosphate. That's where the energy comes in.
Yeah.
Now, in a living cell, and in all the experiments that I showed you, this chemical molecule is abundant. So there is enough fuel to run the reactions. It never runs out. If you run out of ATP, then you run out of the switch, because this change from ADP to ATP and also the hydrolysis, they very much depend on the presence of the ATP molecules. They are the fuel, and you don't change them. You don't change them in the living system, because if you do, the object dies.
There might be a range where then the cell doesn't function, and so
yeah,
I look.
True. The cell has abundant amounts of ATP, so it makes absolutely sure that you don't run out of ATP. So just to give you a number, any one of you has a turnover of 30 kilograms of ATP per day. It doesn't mean you lose 30 kilos a day. That would be nice, I guess, or not so nice, depending on your weight, of course. The point is that it's the turnover of ATP. So it's the amount of molecules that turn over between ATP and ATP. So it's really 25 kilogram turnover. It's a huge amount of turnover in chemical energy. And that's important for your muscles to work, because the motion of molecular motors relative to actin filaments, that's also driven by ATP. And if you were to run out of the ATP, these muscles would not work anymore. They would go into rigor state. Okay, so the point is, you don't want to go down in ATP level. It would be very bad.
Yeah, and this big amount of ATP, you tell us, then this goes, but in a large amount of cells.
In each cell has its own supply. There's mitochondria that are the factories for that, and they make all the ATP supply that you need in the cell. So the metabolism is really, really key. Otherwise, you cannot run any of these non-equilibrium processes. It's a different way of non-equilibrium, right? Non-equilibrium, you know, from things like putting gradients to a system, like a temperature gradient and seeing Rayleigh-Bénard roles. And there the non-equilibrium comes from temperature gradients between the two plates. That's external over a length scale. Here, it's more like a local supply of energy. So locally, for each protein, you have the turnover and a broken detail balance for each of the molecular processes. So it's a local way of maintaining a non-equilibrium state and not maintaining it by boundary conditions or gradients in chemical potentials, temperatures, and other intensive variables.
Thank you. So meanwhile, is there another question by someone? I have to look. No hand risen, and so I thank you again for this nice talk, and I close this session and hope that we see you all tomorrow for the second part and a new topic on these nice days of Ising's centenary. Thank you very much.
You're welcome.
Bye.
It was fun. See you tomorrow.
See you.
And many thanks to the speaker and many thanks to the chairpersons of today's session. Thank you.
Okay, thank you. Bye.